I mentioned to some of you that there is now a contender EU program called the IT Future of Medicine, where both Matthias Ullen and myself are participating. It's one of 6 projects across the board, not all devoted to health, that are competing for funding for 10 years at a rate of 100 million euros a year. So it's quite an interesting initiative, and I think it will take something of that nature, a very large effort to teach healthcare how to deal with the tremendous data flows that we will be seeing, how to model it and understand and compare data generated by whole body scans of PET, for instance, and compare that to transcriptomics and proteomics. And I think our clinicians are far away from being able to deal with that.
So I think one bottleneck actually here, which we're already seeing, is in biology in general, is bioinformatics and computer science as applied to biology. So for instance, in the Science for Life laboratory that we've been talking about, on the Stockholm site, there is more than a third of the personnel is bioinformaticians, although it's a life science laboratory environment. And this is, it's not easy to find enough skilled people in this field, and especially if a project like this gets off the ground. There's going to be a huge demand for people that can bridge between IT and biology and healthcare.
And how challenging is it to get those people to talk to each other in the same language?
Oh, I mean, if you put them in the right environment, that's not a huge issue, I think. And if you bring them in, young people, put them in the right environment, they pick it up pretty quickly.
And that's the purpose of SciLife, to bring people together.
So I don't think that's a huge problem, actually.
I was just going to go back to something you said a little bit ago about the genome project. Back when you're saying it was 10 years ago.
Yeah.
And well, it takes 10 years to develop most, drugs. You'd expect that you wouldn't have got that much out of it, surely. Do we expect that in, say, the next 5 years or so, we will start to see the project actually producing something that could be on the market or close to it? Do you think that's the simple reason for it, is that the time taken to develop drugs? I would just like to hear what Alan's got to say. They think there's going to be maybe a kind of a sudden takeoff in the number of new candidates as the R&D process, starts to bear fruit.
Well, as Lotta is saying, it's here now in terms of monoclonal antibodies. That's the biggest aspect of new drug development. So that's certainly already underway, but it took a long time in coming. Monoclonal antibodies were invented here in England in 1975, or published in first 75. Took quite a long time to get to this stage. For diagnostics, again, it's something that it's been around. I think that will be a piecemeal revolution. It'll come in little increments, but I think it'll pick up speed and develop. But we already have the beginnings of it since far before the genome project. We have lots of protein biomarkers. So I don't think it's possible to say that in 10 years' time this will happen. It'll grow slowly. Some of it is already in place, and eventually it'll grow to a stage where medicine will look very different.
I think to that point, look at Ben Lister, which is approved in the last 2 months, which is a GSK drug partnered with Human Genome Sciences. Human Genome is one of the first companies to start whole-cell sequencing of the genome in 92, 93, something like that. That compound was discovered 10, 11 years ago. It's just reached the market. So I think you're seeing the start of it. But I think there's been a lot, as you said, there's been a lot of basic research that's been done. Now you've got a lot of clinical development work. It's probably another 10 years before we have the wave of genomic drugs. No, sorry. It's probably some time before we get a wave of genomic drugs. Perhaps we could just come back to one other hype in reality. And I want to talk about embryonic stem cells. Because I think it can get people very excited, which means it could be hype. But then on the other hand, we read this week, I think it was at Gothenburg, a young girl got treated with some stem cell therapy, had a serious blood clot, which suggests to me, I don't know if she's still doing fine, I hope so, but sort of that strikes me that's crossed into reality. So where are we in stem cells, and sort of what are the first areas that we could actually see some clinical utility?
stem cells, there is actually a company called Dendrion in the United States that has got an FDA approval for autologous treatment of I can't remember what it was, but
it's prostate
cancer. Yeah. I'm actually very surprised because it actually just increases the lifespan by in average 4.5 months. So I think it's still coming, and a lot of the problems with stem cell or regenerative medicine has been that the methods, tools that the people have been using have not been developed, and that's been a bottleneck before. And we hope that we can actually help solve that bottleneck. But it's definitely coming. There's a lot of evidence. There's a company in the United States called BioHeart that has been treating heart patients from bone. I mean, bone marrow treatment is actually also cell therapy, and that has been used for ages. So I mean, it does work, and it just the embryonic stem cell development has not come yet. Geron, a company in the United States, just got has just put first patient in a spinal cord injury treatment. They had big problems with the FDA in getting this clinical trial approved, and they actually got it approved, and it was withdrawn because they had some safety issues, and then they got it approved again after answering the questions. So it has been, I think, the application that they sent for a phase 1 study was 22,000 pages long, which is a lot, and mostly was safety, and that's because it's new. The tools are not there, but it's coming, and I believe it will work.
I think it's really interesting to compare the protein science and the stem cell area. What I see now, and sort of we both sort of try to contribute to development of new stem cell technologies. We want to provide new technologies and new reagents, and you as well, to assure quality and safety for those drugs. And I think it goes back about 20, if you compare protein science 20 years ago, we're probably at the same level with stem cell therapy today. Still so many tools that are lacking, sets of controls, equipments. It's still very much sort of a research environment, and it needs to transform into more industrial setting. Yes. So I think, again, sort of, it's hard to use a number of years now, but it's probably the same time frame required before stem cells is real. But I think for the proteins, they are real because we have all the tools. They are now sort of commercialized, and it's very robust and safe. Because for every drug, it's safety that's the primary issue. And that's what you need to address first.
I totally agree with you. And actually, I was just in a stem cell meeting before I came here, and one of the sessions was about what can we learn from the monoclonal antibody development, because basically that's what's happening in the stem cell field.
So if I compare again, 20 years ago, in the biopharma industry, we used to grow cells in a couple of liters, maybe 10 liters or 20 liters, which was sort of large volumes at the time. Now we're growing them at 12,000 liters, very industrial settings. And I think we can transfer that knowledge to the stem cell environment, to that community, how we use the technologies to industrialize the manufacturing process. Because stem cells today is sort of produced at small scale, not very industrial technology used, and you also, everything needs to scale up. I think one of the first protein-based drugs was produced in roller bottles, small bottles that sort of roll around. And the way to scale up at the moment was to have more of these small bottles. So you scaled up by the number of bottles. And now that manufacturing process has moved into one big bottle instead, so a couple of thousand liters instead of 1,000 1-liter bottles. And that's the same transformation that the stem cell technology needs to go through.
Perhaps something that sort of was touched on, sort of economics have a bearing upon this. But also, is there a risk that the research is going so fast? And if I look at sort of the chaps I was at university with who are doctors today, and I don't think we even had a computer sort of when I first went to university, or it was a Unix system or something like that. Is the challenge that you're running much quicker than A, the medical profession can keep up with, and B, that the general population, here's your proteome. Well, how do I interpret that, either as a doctor or even as an individual? Is that a challenge for some of these things, making them a reality?
I think you have a very important function in informing the public, and we certainly don't want people to be scared about the prospect of, in my case, diagnosing diseases more readily. So a dialogue between science and the industries working on this with the public via the journalists is very important. So I think that's one of the outcomes, hopefully, of this event, to inspire and enthuse the public about this possible development, which I think could be have a great impact on public health and public spending on health.
But it is a big problem. So just last week we had an event at the Royal Swedish Academy of Sciences where we had people from the Swedish Parliament visit, and we gave some presentations about what's going on in life science. And afterwards I ended up next to two guys who were both fresh members of Parliament, 30, 35 years old. And one of them had some idea about basic science and so on. The other guy was actually turned out part of what's called the Gene Ethics Committee in Sweden, which basically has to come up with rules about what you can do and cannot do from an ethical perspective in gene technology and basic research. And he said, it's I don't know, I have no clue about genes or DNA or whatever, and I just find it a bit odd that there are some guys on this committee that say no to everything. And so I really must learn a little bit more here, because I don't think I should say no to everything. So probably there is a few things that are still okay. But basically he didn't know what DNA was. That was the level of, and this is an important regulatory committee that's run by the politicians. So I mean we really have a big mission there, I think.